Good morning. It's it's a great pleasure to be here. I think what you just heard was, since I speak a little bit of Hebrew, when you want to complain about me, be careful. I was once in a Chinese restaurant, and they complained a lot about the tip we left. We hadn't left the tip yet. We were going to leave a cash tip, and we didn't write one on the receipt. And they started complaining about us, and I said, I speak Chinese, and they almost fell over. So I'm going to talk about gene discovery and also precision medicine. I saw that Abdukia's talk is on splitters and lumpers. This is going to be a splitters perspective. Precision medicine, by definition, is splitting. I really appreciate being invited here. I want to thank the organizers. I was here a couple of years ago, and we went to the medical services area for the Bedouins. It was one of the most fascinating autism-related visits I've done. Chava's not shown, and Mazel's not shown either, but I want to thank all the organizers for this great opportunity today. And you'll get this joke quicker than anybody else, but I showed this slide a couple of years ago at a meeting where biotech was claiming they were very interested in getting involved in neuroscience, and the criteria was genetics first. And I said, you missed the opportunity about over at least 10 or 15 years, because there's plenty of genetic findings. And I said, here's a syndrome you've never heard of before. It's called Mazuyev syndrome. And the cause, I said, is unquestioned. We know exactly what the cause of the disorder is. There's an easy and accurate test for the syndrome. There's high conversion, so if you posit them on the test, you'll likely get the syndrome. There are cell and animal models of high construct validity, so you can recreate the syndrome in animals and so on. There's good preclinical evidence, at least, that reversing the deficit will change the phenotype. There's reasonable estimates of prevalence that put it in the rare disorders camp, the orphan disorders, which is a very valuable place to be in terms of biotech. There's family foundations with this syndrome, Mazuyev syndrome, which are very engaged and will bring you families right away. There are multiple expert sites doing phenotyping in this new syndrome you never heard about. The FDA is very supportive in the orphan disease space, and there's also a reasonable path. If you find a treatment in one of these disorders, there'll be other ones kind of like in Mazuyev too, I said. So they didn't speak Hebrew, and they were all very excited, and they said, I said, if I show you this syndrome, would you get behind it? And they all said, absolutely, this is perfect. This is exactly what we're looking for. I hear some intakes of breath. All right, so I'll come back to that at the end. I'm talking loudly because I don't know if the microphone's picking up, but can everybody hear me? Okay, good. All right, so this is a slide I always use. It basically says that when we think about genetics, it's good we find it useful to divide the space into the frequency of the variant. So genetic variants can be very common or very rare, and they can have tiny effects or very large effects. And just so you understand, this is the area where you won't expect to find anything in autism, because these are common variants of major effect, and autism is associated with a vastly reduced reproductive fitness, so natural selection is at play here. This is where you find ApoE in Alzheimer's disease, because Alzheimer's happens after reproduction. Down here, you're looking for common variants, rare variants of tiny effect. We're not going to find them. They're both rare and have small effect sizes. So the space where you're likely to find variants in autism and neurodevelopmental disorders is this over here. This is where sequencing takes place, and this is where GWAS takes place. And interrupt me if I'm not clear. So a while ago, using a very kind of unique cohort from Sweden, we're able to show that most of the risk for autism is actually in the common variants, those things that have tiny, tiny effects. And everybody in this room has common variant risk for autism. Okay? In fact, common variant risk for autism in the typical population is associated with higher IQ, greater educational attainment, and so on. All the findings I'll talk about are in this tranche over here. These are rare variants of major effect, where each variant can contribute a large amount of the risk for an individual. So again, this is where GWAS is, genome-wide association studies. The biggest GWAS just came out last year. We're going to have about double the samples this year, and there are the first findings of SNP associations for autism, where each one accounts for a tiny, tiny bit of total risk. And I can go into that, but that's not the topic today. So I'm going to focus on these very rare gene mutations that increase risk by many, many fold. So they're kind of Mendelian in the sense of Mendel. So Don mentioned the ASC, the Autism Sequencing Consortium. It's been a lot of fun. It's 10 years last month. There are 50 contributing sites. It's an unusual collaboration because to join, you had to be willing to share your data and or your samples before publication. Usually, most consortia work, you publish your own work first and then you put the data together. We thought that would delay things by at least another 7 years. And what you can see is all throughout the world, people were giving, first they gave data. So first everybody who had data provided it, and then when we had opportunity for sequencing, everybody would kind of go through all their samples and send them to the ASC. This was a paper that came out in 2014, another one in 2015, and we're kind of down here now. All right, and just so you know that Ben-Gurion University is a contributor, which you probably already know. So I'll talk about a paper that came out 2 weeks ago, which we call ASC2, because it's the second major paper. And this is the summary page from Cell. Don't step away from the microphone. Good advice. All right. I can always bring the microphone with me. So what you see here is what we call a Manhattan plot, because it's supposed to look like the skyline of Manhattan. And basically it has the chromosomes, all the chromosomes of the human genome, except the sex chromosomes are not shown here. And then there's a p-value, log of 10, minus 10 p-value, the evidence, strength of the association. And what you can see is there's actually 102 genes that are associated with autism at a genome-wide significant level so far. And I think what's most important is if you look at the top 20 or 30, any one of those, I'm going to jump ahead and give you the punchline, would probably meet criteria for Mizuyev syndrome, right? All right. So I'll go through that paper a little bit. This is to show you that the autism genes fall into basically, they're divided into 2 dimensions here. One is kind of in the top purple that looks green, looks blue here, are genes that are involved in gene expression regulation. So these are genes that modify the effect of other genes. And they're very important in early development. And then this, which is a little bit green on these 2 slides over here, those are genes actually involved in synaptic function or nerve terminal function. So we kind of expected these. These were unexpected, these GER genes, what we call. The other way they're divided is genes that are more likely to be mutated in autism and genes more likely to be mutated in more severe developmental disorders, but also in autism. So this division between the so-called GER genes and the NC genes we knew about already since 2014. And one hypothesis was that these genes over here work by regulating these genes. So because our sample's gotten so big, you can't really see it, but we were able to exclude that hypothesis. In other words, the GER genes cause autism in a separate way than by modulating these genes. And we try to capture that very subtly in the title. So functional changes are the NCs. The synapse or the neuron is functioning differently. And developmental changes is supposed to capture these GER genes that probably change the way that the brain is put together. If you go to Twitter, and I always recommend you don't go to Twitter, look for yourself. Somebody else pointed it out to me. People scoffed at that title and said it's so trivial. It's a neurodevelopmental disorder involved in neuronal function, so of course they're functional. Anyways, that was what we were trying to capture there. But the important thing from this study is that, it's really this thing over here, you can show that the GER genes are not primarily regulating NC genes. The other thing we can now do with single cell data is ask whether autism more maps to excitatory nerves or inhibitory nerves. And what's the answer? Both. Yeah. So if you look at the excitatory lineage, everything red here, lots of evidence for autism genes in the excitatory lineage. Inhibitory lineage, lots of evidence for autism genes in the inhibitory lineage. Right? So Autism is heterogeneous clinically, but also etiologically. On the plus side, as we get more and more mutations, we can begin to understand more and more about how they're functioning. Is it a loss of function? Is it a gain of function? That over there shows you for various genes. Let's see if I can show it. Yeah. This is how many loss of function mutations per gene, and this is how many missense. And what you can see is some genes only have missense, and some genes only have loss of function. So here's a gene that only has missense mutations. So it's called F1, and what you can see is there are lots of missense mutations either in autism or in more severe neurodevelopmental disorders. They're all clustered on the SAM domain, which is involved in dimerization and DNA binding. And so you can begin to understand that this is not a loss of function mechanism. It's probably either a gain of function or a dominant negative. Right? So we can begin to parse out exactly what you need to correct for a specific gene. All right. The other thing that's been really fun is, I think we all know about copy number variants. So a lot of disorders, and I think you'll hear about one later from Doron, 22q11, DeJour syndrome, Williams syndrome. These are recurrent copy number variations. And some of them, probably multiple genes are involved. Some of them, maybe one or two genes in the interval that are contributing to most of the risk. So because we have so much sequence data now, we can say with some certainty that 2q37, a primary driver for the phenotype, is this gene HDLBP, because we see mutations in that gene without a CMV. In 11q13, what's really interesting is that two genes emerge, both of whom are showing mutations in autism. So that's kind of fun. And then I'm going to go back to this question of NDD, or more severe developmental disorders, and talk about, there are multiple studies. This is just the biggest one. This is so-called Deciphering Developmental Disability Study. So in the UK, over the course of about 2 years, every genetic testing laboratory enrolled every patient that came in serially ascertained way for genetic testing, and they collected 12,800 trios over the course of 2 years, and they've all been sequenced. And so they put all that data together to find genes that were associated with more severe developmental disorders. So these are kids who, at 1 or 2 years of age, are already being sequenced because they show motor abnormalities, they don't meet milestones, there's language delay. They're not coming up to they're not at the age to get an autism diagnosis, and certainly not an ID diagnosis, but they're certainly on the path, and they're very obviously showing some kind of developmental delay issues. So, with similar kind of plot, and again, well, you probably can't read it, but some of the genes I showed are exactly the same genes that they found here. So one of the questions in the field was, is everything we're finding a so-called ID gene, intellectual disability gene? I'm okay with that. I think intellectual disability is the forgotten disorder. We put much more money in things that are less severe, including some kinds of autism. But some people thought that, if all we're finding is ID genes, maybe we're not being careful with our resources. So we took all the genes that we found in both cohorts, and we first did a heterogeneity test, which is in the supplement. We said, are these two cohorts different statistically based on the distribution and kinds of mutations in these genes? And the answer is, with a very, very low p-value, yes. They are not the same. The NDD cohort and the ASD cohort are very different statistically. So then we split the genes kind of down the middle. We said, and it's not we don't pretend, maybe this is easier to see, we don't pretend that this gene over here is definitely in that group. This is a binary split, which is a kind of a secondary analysis. But we asked these genes over here, which are more prominent in autism, what do we see different in individuals with mutations of this gene versus the genes that are more prominent in autism and or NDD? Okay? And not only is the mutation rate different, but these are only individuals with autism, so they're in the ASD cohort, but they carry either mutations in the ASDP, the predominant genes, which is this red line over here, or mutations in the blue, the ASD-NDD genes, so the ones that are also found significantly in NDDs. And what you can see is the IQ shifts to the left, goes lower when you have an NDD gene versus an ASD predominant gene, and the age of walking goes to the right. So even within autism, depending on which class of mutation, which gene you have different phenotypes. You're also much more likely to have an inherited mutation if it's the ASD predominant gene, because they are less impactful, right? So mom or dad could be transmitting it, while for the ones that are found both in NDD and ASD, it's much more likely to be de novo. All right, and an interesting thing is that even if you take all the individuals who have IQs above 70, so nominally don't show ID, or above the average of the study, 82, there's still very, very high, very strong evidence for loss of function and missense mutations that we haven't found yet. So even in individuals with autism with higher IQs or typical IQs, we are going to find the same kinds of genes. So that's kind of the summary of the paper. Okay. Any questions about that? So ASC continues to, we try to remain gainfully employed. One of the initiatives that's going on right now is SPARK. So the Simons Foundation is doing kind of a national collection using online recruitment and saliva kits sent to the people. It's called SPARK. Currently, and all the SPARK data is being made available to ASD and other groups like that. And so basically, we are doubling the number of cases right now in the ASC by including the SPARK data. The SPARK data was generated in slightly different methodologies. So we've had all the data for almost a year, less, but it has to be, there's been repeated rounds of cleaning because it doesn't quite meet, other standards. So that's the total sample now in what we call ASC3. So this is provisional to be sure, but this is the ASC paper that I just showed you, plus the new SPARK data, and it has 184 genes. Some will fall out once we do data cleaning, some will come back up with data cleaning, but it goes, we're getting a very significant uptick in the number of genes for autism. And this has really been driven by Kyle and Jack and their various mentors. So one question we have is, are we at a plateau of gene discovery? And so there are all the papers from the ASC. 2019 is the Cell paper, which was filmed by Archive, and that's ASC3, and we're still using either criteria in a linear range of gene discovery in autism. So we can double the sample size again and continue to find genes. And so we're still collecting samples, we're still sequencing them. We're running out of money for the current sequencing pipeline. We hope to get that renewed. But right now, as I'll show you, if this is the paper you just saw, all of these are in process now, including some more from Ben Gurion University, and some other people who very quickly are trying to get us some more samples for the 2020 queue. But SPARK continues to collect more samples. So we do expect that, maybe in 2 years from now, there'll be another 50% increase in total sample size, at least. The other thing that's going on in the ASC is trying to also not just look at the autosome, but to look at chromosome X, which turns out to be somewhat of a thorny problem, but we have some insights now on how to do that. Look at inherited variation, which again is harder to do because de novo mutations are such a good flag for gene discovery. And putting together the SNPs, the common variation and the rare variation. So the ASC work groups are trying to develop new methodologies and new analytical approaches to actually find other sources of risk for autism that are not dominant. All right, so this is some of the work group. They've been terrific. Kyle was the one who made the slide, so he left his own name off, so I put him back on. He's really an amazing guy. And I'm going to switch now to precision medicine. Like, what can you do with some of these gene mutations? All right? And precision medicine, is this idea, at least in the context of genetics, that if you have an etiology, in this case a gene, then you can model that in model systems. You can understand pathobiology. You can think about drug discovery and development. You can think about novel therapeutics, and you can put them back in to the individuals who are affected. And we feel at the CBER Center that we can cover some of that ground, not all of it. We think we can do a good job at saying these are the most important targets for today. We can certainly make human neurons from individuals, participants in our studies or other studies. All of us can make genetically modified rodent models, and then we can also see lots of patients, both within clinics to map out phenotypes, but also within the ASC for gene discovery. We work on 4 syndromes, and this is that same Manhattan plot. So you see we chose the ones that are more frequent and have lower p-values. ADMP, SHANK3, and FOXP1. DDX3X is the work of Dorothy Grice in the back. That's on the X chromosome, which I don't show here. All of these account for, none of these samples are completely ascertained in a completely epidemiologically valid way. So let's say it's 0.5 to 1 to 1.5% of autism and or ID, most commonly both. So they're rare, but they're the most common of the rare mutations. And the p-value not only tells you how many times we found it in the ASC, but also tells you something about penetrance. One of the things we look at is, do we ever see a mutation in this gene in controls? For all those genes, we never do, right? In 100,000 controls, we don't see a mutation in these genes. So their penetrance is very, very high. The odds ratios we estimate in the order of around 50 to 70 to 80. In other words, if you think of the baseline rate of autism as 1%, a 50 to 70 to 80-fold increase in risk, you're coming pretty close to a Mendelian disorder. So they're the most common, that top trunk, and they're highly penetrant. So I'm going to talk about SHANK3, that one over there, and I'll talk about it, very quickly from these 4 kind of perspectives. So first, is it a valid target? Well, clearly it is, but right here it's already a valid target. But even before this came out, we already knew it was a valid target. I would argue that we knew about SHANK3, or at least that region, for almost 50 years, right? So when chromosome 22 makes a ring, you lose genetic material at both sides of the ring. So in the 50s, in the 60s and 70s, you could see that. So that was the first entree into genetic disorders that you could see, because the karyotyping was sufficiently crappily, even the crappy resolution, you could see the circular ring chromosomes. And here's a study of many cases of ring 22, and they have low birth weight, feeding problems, late milestones, low IQ, verbal language delay, et cetera, et cetera, et cetera, et cetera, and they have autistic traits. So even in the 70s, we knew that something about chromosome 22, the very ends of it, were associated with developmental disorder. Here's an individual. There's just 2 papers just came out about this kind of psychosis, atypical bipolar, atypical schizophrenia in this disorder. It was actually also known for decades. So here's an individual with ring 22, has this, he flips between aggression and kind of depression and mutism. There's this rapid cycling, kind of almost bipolar-like traits. And this was really in the literature for 30 or 40 years. Now we get to the new technology of being able to see small deletions. And sure enough, you see the same phenotype when just the bottom part of chromosome 22 was deleted. People began to map this out, all right? And these are big deletions to smaller and smaller and smaller and smaller. And they all show the same phenotype. This paper came out in 2006. This is when we all started sequencing this gene, okay? That said, hey, we keep on seeing amongst all these different size deletions, the smallest interval includes SHANK3 and 2 other genes. And not only that, but there are recurrent deletions in SHANK3. So this was the first paper that said SHANK3 is probably the cause of all that massive syndromes. And that's why, even though there's only an N of 1 in this paper, the people in the business believed it, right? So this is the paper that really kind of put SHANK3 on the map for many people. There's a de novo deletion, a 20q3 deletion, which we've seen literally in the literature, hundreds and thousands of them. Hundreds in the literature, thousands otherwise. This is a balanced translocation, okay? So again, we'd seen that before. And this is the single example of a point mutation, and the first example of point mutation, SHANK3. And this is what made people more broadly aware of SHANK3 as a cause of autism. Now, like I showed you in the ASC paper, there's lots of data with point mutations in this gene. I'll come back to this at the end, but I'll just kind of go through it very quickly. Dad has a balanced translocation, right? So there's chromosome material swap between two chromosomes. That generates monosomy, disomy, and trisomy in children. We have probably in literature have about 10 such families. More outside the living. So disomy is the typical two copies, everything typical development. Monosomy is Phelan-Mcdermid syndrome, very severely affected. Trisomy, 3 copies of 23q13, they have Asperger's and behavioral manifestations. Right? So there's 2 things about this which are, the most important thing is that you can't, for almost all of these genes, too much is also not great either. Too little is bad, too much is usually less bad than too little, but you know, it's a U-shaped curve. All right, so we have our target, and we've seen at the Seaver Center over 100 individuals with this disorder. Everybody who walks in gets a blood draw. All the bloods are cryopreserved, and about 10 or 15 of them have been turned into iPSCs. I won't go through the paper in great detail. It's in review right now. But basically, this is 7 individuals with Phelan-Mcdermid syndrome and 6 sibling controls, and we see some differences in gene expression. So we now have, if you like, you know, from the point of view of drug discovery, we can put these cells in a dish, look at these phenotypic changes, and screen drugs for drug discovery. In terms of genetically modified rodents, there are no less than 12 genetically modified mouse models for Phelan-Mcdermid syndrome. We made 3 of them. Don't ask me why. It's actually a good reason. So this is a summary table of all the different studies that were published at the time. And you can see that some things like repetitive behaviors in red are elevated. There's some deficits in social behaviors. Blue means down. So there's some consistency across the model systems. I'll give you a couple of examples from our work very quickly. The first animal we made was a small deletion, and we saw differences in electrophysiology and behavior. Because of another study that was going on in Rett syndrome, and because of some of the changes we saw in the mice, we tried IGF-1. IGF-1 was known to actually increase the postsynaptic density material in mouse models. And so wild type is a typical mouse. This one over here is SHANK3 mutation, single copy, and this is with IGF-1 IV for 2 weeks. So the IGF-1 reversed the deficit. Another reason we tried IGF-1 is because we knew we could put it into kids very quickly, because it was already a safe drug. It also reversed the motor deficits, which I won't go into. And from another study done by Hala Nicholas, who is actually from Israel as well, together with Shlomo Wagner and her collaborators, we made a rat model where you can do a more complex behavioral studies in rat models. And she showed that the animals have 2 major problems. One is they don't remember a conspecific the next day. This is called long-term social recognition memory. So if you put 2 rats together for an hour, the black and the white one, and then you separate them for a day, they will remember who they met yesterday, and they'll go to the new one. Right? So if you look at the, well, I don't show you the wild type here. What I show you here is with and without oxytocin. So the heterozygote, all the way on the left, shows no preference between the black and the gray. It goes to both rats equally the next day. If you would be given oxytocin before the study, it remembers the black one that it met yesterday, and it spends all of its time with the gray one. So that's social recognition memory. And this is the other thing, which is they show attentional deficits, which I think the clinicians will tell you are very, very obvious in the patients as well. And here too, if you give oxytocin, you increase the number of correct responses, you decrease the number of emissions, and so you're improving. This is the first time oxytocin has been shown to improve attention. And I'm telling you all this because we could take those findings and go into patients. So as I said, we've seen over 100 patients with Phelan-Mcdermid syndrome. And early on, we did the first prospective phenotyping of the deletions. That's what we had most of in those days. It was the first time anybody sat down and prospectively, like, brought individuals in with this mutation and then looked at them deeply phenotypically. You can see that the majority met criteria for autism or autism spectrum. And you can also see that they typically had severe to profound intellectual disability. The point mutations were just becoming a thing, and at some point we said we have to also do the same phenotyping in point mutations to make sure that most of the phenotype is primarily mapping to the single gene, not to the deletion. So we collected over time lots of individuals with a point mutation. We did the same kind of phenotyping. We also added things like EEG. So this is the visual evoked potential. In gray is a typical VEP for reversing checkerboard. And in red is Phelan-McDermid syndrome, and you can see there's a profound difference. Paige Seiper, who did all this work, likes to show a slide where she has, I think, 16 BEPs, and she asks the audience to pick the ones who have Phelan-McDermid syndrome, and you get it right 100% of the time. Because the difference between that, with a big deflection at N75 and up at P100, and that, where there's no deflection and almost nothing at P100, is pretty obvious. And it's across all Phelan-McDermid syndrome. So this is suddenly a biomarker that is actually reproducible within a genetically defined condition. Interestingly, and we haven't done enough of this, in a few studies in mice, we see exactly the same deficit in EEG in response to the checkerboard, reversing checkerboard imaging screen. Okay, so based on the study I showed you in the mice with IGF-1, Alex Kalevzon did a clinical trial it was really a safety trial, but it was kind of safety/efficacy, a very small number, an N of 9, showing some improvement on the ABC social withdrawal with IGF-1 in Phelan-McDermid syndrome. There's a replication study which for some reason keeps on not showing up online in clinicaltrials.gov. The second measure that was significant in the first study was RBS, repetitive behavior scales, and that's replicated. The social has not. Still tiny numbers, but based entirely on the work we did in the animals. I showed you the oxytocin study in animals, and so now Alex is enrolling individuals with Phelan-McDermid syndrome running oxytocin trials in a genetically defined subset of autism. As you know, the history of oxytocin in autism is a bit checkered, and the big study called SORAS-B is about to be released. Certainly didn't meet things, I think, on its primary endpoints, but some very interesting things depending on how you cut it. It's not impossible that oxytocin works beautifully in a subset, but you have to find the subset to show that. And so maybe because in the rats, the oxytocin system is actually clearly profoundly changed when you knock out this gene. Maybe oxytocin will be useful in PMS and other subforms of autism. And this is the clinical trial for the epilepsy in Phelan-McDermid syndrome. This is a company called AMO, and they did exactly what we did, but they did it in a biotech setting. They purchased one of the mouse models for autism. They did some ePhys, they showed some epilepsy, and they tried their drug in mice, and based on that, they went to a clinical trial, and we're one of the sites for the trial. So you can, with the preclinical models, come up with specific targeted therapeutics for that subtype of autism. And that's really what precision medicine is all about. Our problem, I think, as in academia, and certainly at the Seaver Autism Center, is that academia can do all of that pretty well, but it can't do two things very well. One is high throughput, like the discovery part of drug discovery. Companies typically run 1 million compounds through an assay to choose a few lead compounds. It's expensive, and it's hard to do with great, precision. Like people like me, when I work at the bench, it works one day, it doesn't work the next day, I say fine, I try it again, it works again. I don't spend a lot of time getting perfectly clean, 0.001% variance in each of my assays. But if you're running 1 million compounds, you have to do that. So we don't do that very well, the discovery part. We also don't do this part over here very well, which is the medchem, right? So we believe that this is where academia and biotech have to get together. And we're kind of pitching this idea now that, we as a field can do everything up top there. And biotech can't really do that so well. They don't typically have access to patient samples, for example. But they need to come in and bring these two other things. All right, so that's the precision medicine idea. And I want to ask this following question, which is there currently any truly viable alternative, right? You might think precision medicine, you know, we're talking about 1% of autism, 0.1% about 0.5% of autism. You know, we all want to treat everybody who needs help, right? And so parsing away or cutting away, you know, 0.5% of autism may seem like, you know, a bad strategy. I would argue that it's the only strategy that's likely to produce results in the short term from a pharmacological perspective, not a behavioral one. Behavioral, you know, we know it works to some degree. So there's a way of what I call top down. You look at the phenotype and treat that, and bottom up, you look at the etiology and treat that. I would put it now, and if Joe gives words, is you lump based on phenotype, but you split based on etiology. So is there any viable alternative? So this is what I kind of hoped to convey. There's not a single pathway in autism. Just forget it. We used to think that maybe there was a couple of genes. That's clearly not true. But even within the 100 genes we have, clearly not a single pathway. We do know, however, that most of the mutations we are finding are dominant. So there's a second allele, which could be targeted. If they're missing something you want to add, you can work on the one that's still there and intact. Primarily loss of function, which means you want to put it back. So we kind of know mechanistically. They can show a U-shaped curve, which is a warning. What I showed you before, if you try and put it back and you overshoot, you may get phenotypes too. They tend to be severe. So, you know, everything I'm talking about is associated with fairly significant severe autism. Right? These are not, you know, what people sometimes call high functioning. These are people who really, really, I think we would all agree, would benefit from intervention. And there are multiple examples. I'll show you one where in vitro, if you put the gene back in, you see a recovery in a mouse model. The genes are, quote, fairly common. In other words, pharma could get interested. And they're highly penetrant, which means that they really cause the disease. This is just one example of no single pathway. This is just the excitatory versus inhibitory, but I showed you neurocommunication versus GDR. Most of the genes have PTVs. That's a loss of function allele. There are some exceptions over there that are gain of function or dominant negatives, but 80% of what we're finding is loss of function. So it's just gone. So if we could put it back, things could get better. I showed you the U-shaped curve over here, but it's also true for all the copy number variants, right? The Williams syndrome region has a duplication, DeJour's has a duplication, and invariably, almost invariably, the deletion is there's a phenotype with the deletion, there's a phenotype with the duplication, and generally, it's more severe with the deletion and less severe with the duplication, but it's all this U-shaped curve. Tend to be severe, intellectual disability in the severe to profound range, significant autism, very severe sensory problems, a lot of anxiety, a lot of attentional deficits. There's a lot going on here. This is one of the papers where they knocked out a gene, let's say SHANK3, and then they turned it back on at different stages of development, and they showed a very extensive, not perfect, but a very extensive recovery in the mice, even if they turn it back on in adulthood. There's a caveat, a paper just came out that talked about this entire class of experiments maybe having something we don't control for perfectly, but there's probably, for the top autism genes, there's probably 10 of them have been shown that if you turn them back on in later development or adulthood, you get some reversal of deficits. And then I said also they're relatively common, 1%, 0.5%. Okay. And then I also make the point that the odds ratios for the top tranche is 20 to 70, so very significant contributors to risk. So that's kind of what I tried to show you here. And let's come back to Moussouaf syndrome. So if I told you that SHANK3, or Phelan-Mcdermid syndrome, which is its other name, the cause is unambiguous and uncontested, you'd probably agree, right? Everything we know about Phelan-Mcdermid syndrome is 99% of the phenotype is mapped to one gene. There's an easy and accurate test. It's called a genetic test. High conversion. We see no such mutations in 100,000 controls. We're pretty sure that SHANK3 heterozygous loss of function or deletion is fully penetrant. It's going to produce a phenotype, maybe not autism every time, but certainly ID in the vast majority of cases. So high conversion. It's easy to knock out a gene or a locus in a mouse or a cell. We even have cells from patients, so we have very good model systems. I showed you that slide, previous slide. This is actually, as I said, Shank3, okay, where if you turn the gene back on in mice, you reverse the deficits. So that's right over here. All right. Reversing the causal deficit will lead to improvements, even if given later in life. Unbiased estimates, maybe it's 1 in 20,000, maybe I'm off by a few percent, but that's very, very much in the orphan disease space, which is very valuable commercially. I hate to put it in those terms, but that's all pharma wants to hear. That's not true. That's the first thing they want to hear. If you convince them of that, we can move on. There are multiple family foundations associated with the rare disorders. Phelan-McDermid syndrome has a family foundation and a new foundation called Cure Shank, only focused on drug discovery and development. There are many sites. I showed you the Seaver Center, but the Seaver Center is part of a network of 7 sites in the US. It's part of an international network called ECHO. So there are many sites where you can do a clinical trial or you can get phenotypic information. And the FDA is truly very supportive in multiple ways we can talk about, not only because of the orphan disease and the unmet need, but because they failed so miserably with some of these disorders that they're very positive about trying to work with companies to come up with the indication. And then it's almost certainly true that if you find a treatment for one rare subtype of autism, then you could probably try it in a few other, and it might work, like in cancer, right? All these new cancer treatments, they try them in one thing and they immediately pivot once they're approved and they say, oh, it also works in. All right, so this is the punchline for Takeda, but you already got the joke. There's no such thing as Mcdermid syndrome. Mcdermid is Hebrew for fake. However, there are many NDD syndromes with most of these properties and a number with all of them. And the reason I only have most is because we haven't looked at it yet. The fraction that have all of the properties I showed will increase as we do more studies. So from that perspective, I think drug discovery in severe autism is going to almost certainly need a precision medicine approach. And I will stop there. So thank you for your attention.
Thank you very much, Joe. We have time for 1 or 2 questions.
Okay, my name is Shmuel. During your lecture, you spoke in generally about medicine aspects of precision of autism, and I want to ask you if research of precision of autism, including social aspects of autism, this side influence on autism of social problems, of poverty, of life environment of autists.
Yeah. So I have to be very clear that we're talking about, pharmacotherapies in severe autism, and it's not to replace any kind of social or behavioral or other intervention, right? There's no question that that's actually, we can argue how good it's going, but it's certainly going much better than pharmacotherapy. So just like with other disorders, like with OCD, you can treat it behaviorally, but you may want to also treat it pharmacologically, right? So I think that there's a place for both. I don't think this is supposed to put aside any kind of social intervention, any kind of, other environmental causes of autism or contribution of autism. It's about particularly the more severely affected and bringing them up to a point where the social and behavioral interventions can be much more effective. Does that make sense? Thank you for your question.
Yeah.
Thank you for a wonderful lecture, as always. I have a small remark, maybe not really a question, about the ascertainment of prevalence of those 4 disorders that you showed. Yeah. And I believe at least for 3 of them, there is some referral bias for SHANK3, ADNP, sure. And DD3X, because they have so many additional features besides autism, intellectual disability, dysmorphic features, and also the pathogenicity that you showed. Maybe it causes that not all those fetuses are produced, because in fact we see much less than 1% of those disorders. And unlike FOXP1, which is, it doesn't have any additional features, and autism is more like high-functioning autism, so maybe the ascertainment is better in this disorder.
So I have refused to give numbers for many years when asked, but I'm always asked. And at some point, it became counterproductive not to at least guesstimate, because, like as I said, biotech or pharma won't even start the conversation unless the Family Foundation could come up with a guesstimate. So I give my guesstimate, and I'm trying to find a slide, but maybe I'm wasting everybody's time. It's heavily biased towards the DDD samples that were actually serially ascertained, but of course they're biased samples. You don't get, in the UK, you're not referred to genetic testing unless you already have a fairly profound phenotype. But even within that tiny group, the rates of SHANK3 are quite high, ADMP quite high, even higher than in the ASD cohort. So you're right. I think it's probably a factor of 2 either way, probably in the rarer way. But it's probably not off by more than a factor of 2, and we just don't know it until we have universal testing. Or we could do a truly national epidemiological study, which we can't do yet. But absolutely true.
Okay.
Okay.
Thank you
Thank
again,
you.
Joe.